So I'm humbled by this opportunity to speak about chemical proteomics and dry design, because I'm a physicist by training, and a significant part of my career I was building mass spectrometers. And only recently I started to get involved into the research area related to drug design. And that was when I joined Karolinska Institute back in 2009. And of course, Karolinska is about medicine and drugs and diseases, because Karolinska, as we know, is charged with awarding Nobel Prize in medicine or physiology. So there is also a great need to discover new therapies, because as we know, current methods are not extremely efficient, especially in oncology and Alzheimer's. And these are two areas where we are most active. As drug targets, today there are mostly proteins that are playing this role, almost 94%. And that's for a good reason, because proteins are doing things. And many proteins, about half of them, have enzymatic activity. Perhaps if we include moonshining protein, then there will be more than half of all proteins. But in principle, every protein can be a drug target, because every protein is there for a reason, and it affects some important biological processes. On the other side, there are small molecule drugs, and the chemical space, as you know, is vast. It's as vast as the universe. And there's so many new molecules to discover. But of course, as somebody already said, a good drug, I think Roman said that here, the good drug would combine many different properties, including solubility, low toxicity, availability, and so on and so forth. So the way how most drugs work, they go into blood from stomach, and then they go around the body, and they find its target protein, and they bind allosterically or orthosterically, and that's how they block typically the action of the protein. The problem is that most drugs are not really specific, and even the drugs which we touted as being extremely specific, like the silver bullet supposedly against cancer, Gleevec, we're supposed to bind only to its intended target, tyrosine kinase BCR-ABL, but in fact, even Gleevec has many different off targets that are engaged at different concentrations. You see on this slide, the larger the bubble, the lower is the concentration at which the drug engages its target. But basically what you see is that most drugs, if not all, they bind to several proteins. And the question is, of course, what proteins can they bind to, interact with? And that's the questions that we're trying to answer. And recently we were able to answer the question how long the drug and protein target stay together in vivo. So we all do that with chemical proteomics. And chemical proteomics is this: we have a facility, and people come to us with a new molecule, and then we do cell work, we do mass spectrometry. And at the end, we will present our collaborators or our customers with what we think is happening. We present the candidate for target, the binding site if they need it, the mechanism of action. If it is an anti-cancer drug, we tell them the mechanism of death, the changes in the redox state, and the time of protein drug complex inside the cell. So how do we do that? Well, we do that all with mass spectrometry. And what is mass spectrometry? It's just a way of weighing molecules in the gas phase. So this slide is a little bit complicated. I would like to ask for your attention for a minute when I explain it. It may be of interest for you to know the inside of the proteomics. It starts with tissue or cell or bodily liquid, which from which we extract proteins, and then we digest them. Well, upon reduction and alkylation of the sulfate bonds, we digest them typically with trypsin, and then we get a mixture of tryptic peptides. And more often than not, we label each peptide with so-called tandem mass tag. That's a small molecule designed to be attachable to free amines and N-terminus, for instance, so lysine side chain, and it contains two parts: the so-called reporter region and the tail. And when then later peptide will be fragmented in the gas phase, this reporter part charged will be released and give a peak in the mass spectrum, which will tell us the relative abundance of this peptide. So this tag is designed in such a way that the masses are
in progress.
The masses are different because of isotopic labeling, but the mass of the whole tag is the same. So one can multiplex up to 18 different samples into one mixture. And then so one can label the whole experiment with sample, controls, treated, untreated cells, as well as replicates into one single LCMS analysis. And then peptides are separated by chromatography and ionized by electrospray. And here is a mass spectrum that emerges, and the instrument looks at every peak here and figures out, looking at the isotopic distribution, it figures out the charge state, and then decides that this may be a peptide, and then it undergoes fragmentation, and then you get another mass spectrum, tandem mass spectrum, MSMS, using one of 3 available techniques for fragmentation. And then a software will match this to the database to identify the sequence of the peptide. And then we have these TMT reporter ion regions that tell us relative abundances of the peptides, that is, relative abundances of that given peptide in a mixture. And at the end of the day, we identified and quantified proteins in a number of samples. We can do actually more than 18 experiments at a time, but we need to use several TMT sets and then merge the data. So it all sounds very complicated, because it is. There are a lot of work has been done to make it work. At the end, we get either a volcano plot with a difference on the X scale and the logarithm of the p-value on the Y scale, and anything above the threshold is of interest to us. Or we use OPLSDA, kind of a supervised principal component analysis, where we contrast the action on the proteome of one molecule against all other molecules and controls. So the proteins that are specifically upregulated in response to that molecule will be to the right, extreme right, and the proteins specifically downregulated in response to that molecule will be on the left. And it's all very nice and dandy, and we apply the same approach to 3 different dimensions of analysis. One dimension relates to the concentration of proteins, we'll call phi-exp. I will explain what it is. The concentration of proteins change, and then another dimension relates to the solubility of the proteins, called thermal proteome profiling. That's complementary. And the third one relates to redox state, which is basically the state of cysteines. And as we don't trust any single dimension, we merge the data, and then we tell by the ranking what is happening, most likely. So we typically would require that 2 out of 3 dimensions would support our conclusion. And then, of course, the residence time of the drug on the protein. Okay, functional identification of proteins. Targets by expression proteomics, so phi-exp. We came up with that around 2015, so it's pretty fresh, as almost everything we are doing. Basically, it's based on the observation that in extreme apoptosis, we're talking about anti-cancer drugs when the goal is to kill cancer cells. In extreme apoptosis, the abundance of the targets is changing dramatically, more than any other protein. And that's how we determine them. To give you a few examples, for instance, there's a drug, methotrexate, and so we apply it to in a panel of 60 other drugs, and then you see the response of the target protein, DHFR, to all these drugs. And you see that the most significant positive change occurs when it is the case for methotrexate, that is, when this protein is the target for that drug. And somewhat less significant for Tomudex, which is also the drug targeting DHFR. That's one example. Another example is the protein TOP1, and you see its abundance goes up and down in response to different drugs. But when it goes down very significantly, it is, in all three cases, the target of the corresponding drugs. Okay, so the same information we can get in OP-LSDA. And so to give you an example here, so when we compare the action of methotrexate against the action of all 54 drugs on the proteome and proteome of the cells, we see that there's one outlier here on the positive side, and sure enough, it is DHFR. And here, paclitaxel, we see upregulated proteins, and these are beta-tubulins. And of course, paclitaxel is known to be a depolymerization inhibitor for tubulins. There's another drug, which is vincristine, which also acts on tubulins, but it's polymerization inhibitors. And then we see the same proteins on the other side, they're downregulated significantly. And so on and so forth. We can see proteasome inhibitor. We see this protein, OSW1, that affects mostly transmembrane proteins and so on. So one can do hierarchical clustering of the drugs and proteins and define the groups of drugs which act similarly. So classify the drugs by their mechanism of action. One can also look at the total proteome change, and with principal component analysis, one can define that there are about 13 dimensions, orthogonal dimensions, at 1% cutoff for in PCA. And that roughly corresponds to 13 known orthogonal mechanism of cell death. So there's a good correspondence. So we published this method in a number of papers, but we wanted to see whether it works also at the single cell level. And single cell proteomics is a very recent development. I think the first paper was published back in 2019 by my former PhD student, Bogdan Budnik, and his collaborator, Nikolai Slavov. What they did, they brought the sensitivity of mass spectrometry to the level of single proteome. That amount of protein is less than a nanogram, a fraction of a nanogram. And so what is the trick? Well, the trick is to use this tandem mass tag and label different single cell proteomes with the different labels. But 2 out of the multiplex samples, or 2 or 1, has 100 or 200 cells. So this is a so-called carrier proteome. And as all these in multiplex samples, the signal in mass spectrometry is summed together. So the mass spectrometry sees it as a proteome of 200 cells, which it can deal with. And the question was whether we can see the drug action and determine the drug target using FITEX method on single cell level. And for that, we used 3 different drugs, one of them methotrexate. And we did see that DHFR gradually rises up in terms of the ranking. After 3 hours of treatment, there is no change. And we could also see part of the proteome of the cells that are going to die, because the concentration of the drug was equivalent to LC50. That is, half of the cells were dead after 48 hours, half not. We were interested in the question, at which point does the cell decide when it's going to die? So we determined a number of proteins which were characteristic for cell death, and then we could see how different cell populations. Here, every dot is a cell, how these two cell populations differentiate over time. After 3 hours, there's no difference. After 6 hours, the difference starts to emerge. But DHFR as a drug target is still not very regulated. After 12 hours, we see it's started to be upregulated, and we see the two subpopulations pretty well differentiated already. After 24 hours, they're well differentiated, and after 48 hours, very well. And of course, DHFR is one of the most significantly upregulated proteins. That's at single cell level. So I can now monitor the drug action in the cells at the single cell levels. But there was a protein abundance. That's one dimension. As I said, we don't trust one dimension very much. We need another dimension, and this dimension is a protein solubility. What is known is that if you increase in the temperature, the proteins would get unfolded and they will lose their solubility a little bit and they start to be aggregating. But if the protein binds a drug, the solubility changes one way or another. And my former PhD student, Misha Savitsky, came up with a method called thermal proteome profiling. Of course, he was working together with some other bright people. Here they published a science paper back in 2014 when they show that with this thermal proteome profiling, or TPP, you can measure using proteomics the melting curve of thousands and thousands of proteins at a time. And then you do it with and without the drug, and you look how this melting curve changes with the drug added. And if it changes, then this protein is interacting with the drug. And that's how you determine the drug target. And that works very nicely. And so you can, as a drug, use a protein. So you take a recombinant protein and add it to cell lysate and see how the proteins shift their melting temperature. And those that are shifting, they're those that build a complex or with that protein added, so that you can determine protein-protein interactions. And that's a very powerful method. It works very well. You can also determine the substrates of enzymes if the enzyme requires both a cofactor. So the most changes, significant changes occur when both cofactor and the enzyme are present. Then you see most significant shifts in the solubility of the substrates. And knowing that, we can design an experiment. And I can show you just one example, thyroidoxin reductase. It is reducing disulfide bonds. So we add this recombinant enzyme together with this cofactor NADPH to the cell lysate and do the experiment. We also add them one at a time and nothing at all to compare, and then everything said and done, then we get these candidates for the substrates of thyroidoxin reductase. And this is just a prediction, of course, so we need to validate it. We always validate our findings. And if the method that we chose for validation is called redox proteomics. And as I said earlier, redox proteomics is looking at the state of cysteines. And usually people say that there is no disulfide bonds inside the cell. So all cysteines are reduced. This is not exactly true. About 10% of cysteines are engaged in disulfide bonding, even inside the cells. But this percentage can go to 20 or higher if a drug is used on the cell that increases the oxidative state, that is, induces oxidative stress. So we label the cysteine specifically with a different label, and then do the experiment and look at the ratio between the two labels, and that's how we determine the state of each cysteine, and also how the state changes. Thermal proteome profiling is a great method, but it has a limitation. One of the limitations is that it requires you to do a lot of thermoproteomics analysis. You need to have at least 2 data points for each melting curve. That means you need to do 10 experiments. And then we thought how to compact this experiment, and decided to measure not the curves per se, but the integral under the curve. And how do you measure the integral under the curve? If the curve shifts, then the integral shifts as well. So how do we measure the integral? We just sum together, pool together all these 10 samples and analyze them at one. And there's no curve fitting anymore. You just look at the ratio or the difference in the intensity of the corresponding ions, and that increases your throughput tenfold. And if you add another dimension, which could be concentration of the drug, then you increase the throughput 100-fold. We call this method PISA. That's an abbreviation. And with PISA analysis, we can also measure the residence times of the drug. And how do we do that? Well, first of all, let's use it in living cells. We take, or it can be used on cell lysate, but actually on living cells, it's easier. So we take a cell, add a drug, incubate for some time, and then split the cell population into a number of subpopulations. And for each subpopulation, with a certain delay, ranging from 1.5 hours to, say, 24 hours, we remove the drug and watch the cells and continue incubating until the rest of the timeline, that is 24 hours. And then we extract the proteome and do the analysis and look, and we do PISA analysis and look how this shift in solubility changes with time. And so we found that different proteins, of course, bind the same drug with a different efficiency. And when we normalize these solubility shifts, we can see this decay curve corresponding to the drug leaving its target. And sometimes it leaves completely, so the solubility of the target goes back to the background level, or sometimes there's a significant shift remaining, that is the drug stays with the target, maybe because of the covalent interaction, or maybe because it's a non-covalent but strong interaction anyway. And therefore, we can rank our targets by their ability to retain the drug. So we can do PISA when the changing parameter is concentration. And in this case, the drug that starts to show, the drug target that starts to show solubility shift at the lowest concentration is the best. Or we look at the solubility shift, and of course, there's a simple-minded idea that the biggest shift the protein exhibits, the more important it is as a target. And then the residence time. The longer time the drug stays with the target, the better it is. And so we have 3 parameters. We have to combine them into one. We do it through ranking, and then we can rank all the targets. And you see, for that particular drug, the protein lean shows the most significant behavior. That is, it's ranked at the top of the list in concentration analysis, and it's one of the most significantly changed protein in 2 other dimensions. The limitations of PISA, however, is that temperature. Temperature is our friend. It's cheap. It is cheap to change temperature. But in some cases, you don't want to change temperature because the interactions are very labile. You increase the temperature and the drug leaves the target and your analysis fails. So we would like to have a similar method, but without change of the temperature. And so, as many people know, you can use salt concentration that does the same thing. You can desalt proteins. So you can salt them off. That is, by adding salt concentration, increase the salt concentration, you see protein aggregation. And we use this in the method called ion-based PISA. Not all salts, of course, are created equal. Some of them are better than the other, and some don't change the solubility of the protein, and some actually increase the solubility of the protein. And we choose zinc chloride, and zinc chloride works like a charm. So one of the side effects of this method, one of the benefits compared to temperature, we can reduce significantly the volume of the sample, and we could go sub-microgram amounts. That's what you cannot do with temperature PISA. But we are still not at the level of single cells. There's still about 3 orders of magnitude to do the improvement to bring this method, PISA, solubility-based method, to the single cell level. And there's a significant correlation between the temperature PISA and zinc PISA. However, there are also proteins on which temperature-based PISA TPP works, but salt doesn't work, and the other way around. Salt works, but PISA does. Temperature-based PISA doesn't work. With that, I come into conclusions. Chemical proteomics reveals drug targets without chemical modification of the drugs. FITEXP method, we also call it ProTarget Miner because we wrote a software for that and we created a database. It's freely available on the web. Uses expression level changes for drug target deconvolution. It is already translated to single cell level. And TPP and PISA perform target deconvolution based on solubility changes. And PISA allows one to determine interacting partners of the proteins, enzyme substrates, and the residence time of the drugs. And ion-based PISA reaches sub-microgram level of sensitivity. There's more to do. There's more methods to create better sensitivity, go into single cell level for all dimensions of proteomics. Thank you very much for your attention, and I will also like to thank my former group member, current group members, and funding agencies. Thank you.
Thanks a lot, Roman. And unfortunately, you almost do not leave time for discussion. Yeah, and I actually don't see now. Yes, there is some question. Maybe I will read this question. Just a moment. So, are TPP, PISA, I-PISA appropriate only for protein small molecule interactions or for biologicals too? There are so many anti-cancer monoclonal antibodies now.
Yes, that's a very good question. Indeed, we are now working on using PISA for antibodies. And also you can use the solubility change just for profiling biological systems to see how changes in biology are reflected in the changes in solubility and learning something about the processes. For instance, we use PISA together with expression proteomics. We call it PISA Express in profiling stem cells, how they differentiate along the way. And we discovered that one of the ribosomal factors is actually key for differentiation. The blocking of that protein inhibits differentiations of stem cells.
Okay, maybe another question. What does area under the curve in PISA represent physically? I get that when one of the curves shifts, you might get a larger, smaller area for that curve. But what is the physical meaning of taking an integral in these coordinates?
That's a good question, philosophical. I don't know. I don't have a ready answer for that. Your guess is as good as my guess.
Okay, thanks a lot, Roman, once again.